Hi everyone and welcome to today's lecture on lipid metabolism. As always, I just want to remind you that after each slide you will find a slide labeled chalkboard notes for you to pause the video and jot down some notes. So when we talk about lipid metabolism, the first one that I want to mention is fatty acid metabolism. And we've mentioned previously when it comes to lipids and fatty acids that they're very important in terms of catabolic and anabolic reactions or processes. And as a reminder, again, catabolic is when you're breaking something down, whereas anabolic is when you're building something up. And so when we talk about fatty acid metabolism, there's the catabolic purpose, which would be when you have fatty acid oxidation to provide energy for cells. And this would occur if, let's say, the glucose levels are low. And so you don't have the carbohydrates for energy, so you have to use fatty acids instead. Also, you end up with anabolic purposes, and that is that fatty acids can be used to build or create very important biological molecules. And so some of the examples are triglycerides, phospholipids, which are critical in your cell membranes, second messengers, and even some hormones as well. Now, when we talk about fatty acid metabolism, again, whenever you hear metabolism, think breakdown and building up. So, depending on what you need at that time, you'll either have degradation or synthesis. So, with fatty acid metabolism, it hinges on fatty acyl CoA. And think about it, if energy is low, you need energy, right? And so, if let's say you don't have glucose or carbohydrates available to get you energy, then what are you going to want to do? You're going to want to break down fatty acids, right? And so, where do you make energy? Well, think of all the memes that say powerhouse of the cell, the mitochondria, right? So if energy cell charge or energy is low in the cell, then fatty acyl CoA is going to want to enter the mitochondria. And it will do this by what's called the carnitine transport cycle. And we'll talk about that in a moment. And so once inside of the mitochondria, then you can have the breakdown of fatty acids or fatty acyl CoA into acetyl CoA, FADH2, NADH, all valuable things that we've mentioned before that are great in terms of making us energy. Now, sometimes the opposite is true. Sometimes you have lots of energy available. So if you have lots of energy, energy is high, you don't want to break down the fatty acids, right? Instead, you're going to want to do the opposite. You're going to want to build fatty acids because if you have energy, energy allows you to build things. So instead, when the energy is high, synthesis is going to be favored. And you will have a block or inhibition preventing fatty acyl CoA from getting imported into the mitochondria. Okay, you don't want the fatty acyl CoA to go into the mitochondria if you have lots of energy. Instead, you want to go toward synthesis. So something called malonyl CoA will inhibit mitochondrial import of the fatty acids. Okay, so a moment ago I mentioned the carnitine transport cycle. And what we said that was important for is for getting fatty acids into the mitochondria. That way you can break them down and ultimately get energy out of them because this is going to happen when energy levels are low in the cell. So the carnitine transport cycle is basically to then allow to get the fatty acids into the mitochondria to then be able to enter the oxidation cycle, okay, which is going to be a three-step process to ultimately get to break down those fatty acids. Okay, so again, this is going to occur when energy levels are low. and synthesis is going to, you're not going to want synthesis. Instead, you're going to want to break down the fatty acids. That then brings us to the fatty acid beta oxidation pathway, which is a fancy way of saying we're going to be breaking down fatty acids. As I mentioned, this is going to occur when you are low on energy, right? So you want to break down the fatty acids to get some energy. And if you're doing this for energy, that's going to take place where? In the mitochondria. Now, what's going to happen is you will get the breakdown of the fatty acids as carbon pair units at a time. And basically, the ultimate goal is going to get us FADH2, NADH, and acetyl-CoA. Now, for these, I'm going to want you to know that when you're breaking down fatty acids, not only that these are the three products you're getting, but I want you to know how to calculate how many of each you would obtain, depending on how many carbons you have in that fatty acid chain. So first of all, you're going to see some calculations on the next slide. N will be the number of carbons in the fatty acid. So for instance, if you see a fatty acid and you see it labeled as C16, that tells you that there are 16 carbons in that fatty acid chain. And so in our calculations, N would be 16. In order to get the number of acetyl-CoA produced from breaking down that one fatty acid, you would do N divided by 2. So the number of carbons divided by 2. So let's say you had C20. Well, that would tell you that there are 20 carbons. And so the number of acetyl-CoA would be 20 divided by 2, which is 10. So you would get 10 acetyl-CoA produced from breaking down that particular fatty acid. For FADH2 and NADH, the amount that you would get is N divided by 2, and then you subtract 1. So let's say you're breaking down that 20 carbon chain fatty acid. The number of FADH2 that you would obtain would be 20 divided by 2, which is 10, minus 1. So you would get 9 FADH2. Okay? N divided by 2 minus 1 is not only the number of FADH2 or the number of NADH, but it will also tell you the number of rounds or cycles needed to fully metabolize that particular fatty acid. Okay? So I can give you a fatty acid, I can tell you how many carbons it has, and then you should be able to tell me how many acetyl-CoA you would be able to obtain from breaking that down or oxidizing it, from the oxidation of it. You should be able to tell me how many FADH2, how many NADH, or I could even ask you the number of rounds, which the number for FADH2, for NADH, or the number of rounds, that number is the same for all three. Okay? And we're going to practice this in a moment as well. Okay, so here's a practice problem about the calculations, and then we're going to take it a step further and be able to calculate how much ATP we would be able to obtain from the oxidation of a particular fatty acid. So in our example here, we're going to have Palmitoyl-CoA, and it's C16, so that tells us that there are 16 carbons. So if we're thinking of our calculations a moment ago, that would tell us that N is equal to 16. So now, taking this a step further, in addition to figuring out how much NADH, FADH2, and acetyl-CoA we would be able to get from these 16 carbons, we also can figure out how much ATP we would be able to get. So I put a little code here that I want you to memorize. For every NADH that we get during fatty acid oxidation, that gives us 2.5 ATP. For every FADH2, we get 1.5 ATP. And for every acetyl-CoA, we get 10 ATP. Okay, so now we're going to go one by one and figure out how many NADH we would get and how much ATP we would get for each for this particular fatty acid based on each carbon that we have. So NADH, we have 16 carbons. So we said a moment ago that we do N divided by 2 minus 1. So 16 divided by 2 minus 1, that gives us 7. So we'd get 7 NADH. And then if we want to know how much ATP that would give us, well, we have 7 NADH, and for each one, we get 2.5. So 7 times 2.5 tells us how much ATP we would get from the NADH that we just made. At the same time, when we're breaking down that fatty acid, we're also getting FADH2. The calculation for that amount would be the same as NADH. You do N divided by 2 minus 1. So we would get 7 FADH2 from the oxidation of our C16. And then for each FADH2, we would get 1.5 ATP. So you do 7 times 1.5. The last one we have to calculate is acetyl-CoA. That one's a little easier because it's just N divided by 2. So we do 16 carbon divided by 2. From the breakdown of our C16, Palmitoyl-CoA, we would get 8 acetyl-CoA. The amount of ATP we would get from that acetyl-CoA would be 8 times 10, because 10 ATP per acetyl-CoA. So that tells us if we were to break down our 16 carbon fatty acid, you would add together all 3 ATP amounts, and we would be able to get a total of 108 ATP. But we're not done yet, because this process requires the use or taking 2 ATP to activate the process. So for activation. So what you have to do for the final answer of how much ATP we would get from the oxidation of this fatty acid, you take the total ATP, which is 108, and you subtract the 2 that was needed for activation. So our final answer would be, if we're going to break down this 16-chain carbon fatty acid, we would get 106 ATP. Okay? So I could give you any fatty acid. I'll tell you how many carbons it has. You should first be able to figure out how much NADH you can make, how much FADH2, how much acetyl-CoA, which is the number over here. Then you should be able to figure out how much ATP. So you need to do this portion, which is the multiplication portion, add up the ATP that you got from those 3 products, and then subtract 2. Okay, so for instance, a moment ago I had said, what about if we had C20, right? 20 carbon. Well, what we would do is for NADH, we would say how many NADH? 20 divided by 2 minus 1, which would give us 9 instead of 7 here. So we would have 9 NADH, 9 FADH2. We would have 10 acetyl-CoA because it would then be 20 divided by 2. And then for the amount of ATP, we would take our 9 NADH and multiply by 2.5. We would take our 9 FADH2 and multiply by 1.5. And we would take our 10 acetyl-CoA and multiply by 10 ATP. Once we added all 3 of those values together, we would subtract 2 for our final answer. Okay? So I could ask you this with any number of carbons, just like we just practiced with 16 and we practiced with 20. If you're unsure of any of this and want more practice, just send me a message in the Remind app and I'll send you a little more practice. Okay, that then brings us to something that has become quite popular in in gym training or pop culture and everything, which is the term ketogenesis. And that's basically a process that happens when that acetyl CoA that we were just talking about that gets made in the mitochondria when you're breaking down fatty acids. Well, when there is excess of that, what can happen is it can be converted to ketone bodies, which, let me just grab the laser pointer here, are over here acetoacetate and hydroxybutyrate. So if you can circle those two names and recognize if you ever see acetoacetate and hydroxybutyrate, circle star highlight those are ketone bodies. And so ketogenesis converts the extra acetyl CoA to those ketone bodies, and that becomes valuable for that area because what happens is during starvation when carbs are low, so carbohydrates are low, the body can then use those ketone bodies in order to ultimately obtain energy for your muscles. So you see how over here the ketone bodies are sent to muscle tissues where if there's not enough carbohydrate, you're starved for carbs, you can then use those ketone bodies as a way to get energy for your muscles. Now, we're going to look at in some of the other slides a little bit about what can then happen in terms of those and why it's not the best idea to do those keto diets that unfortunately became kind of popular, especially on social media. Okay, so let's say those not so great keto diets that I was just talking about a moment ago. Let's say you put your body into ketogenesis. Well, now what happens to those ketone bodies that were just produced with that excess acetyl CoA? Well, what happens is they're basically able to travel through the blood and throughout the body, and then cells, especially skeletal muscle and in the brain, can convert the ketone bodies back into acetyl CoA. And that ends up being very valuable because that can then enter the citric acid cycle to produce ATP. So you can see that over here, that acetyl CoA going now back into the citric acid cycle, and that then produces nice energy. Now, the problem with diets like this is that because you are on a low carbohydrate diet to kickstart ketogenesis, because we just said it occurs when your body is starved for carbs. Nice little rhyme to it. So, starved for carbs. And so now you're on a low carb, high fat diet. That's going to end up leading to things like cardiovascular heart problems. You're going to have GI problems. There'll be a lot of lightheaded, kind of dizzy, low blood pressure issues. You could end up hypoglycemic. And what's interesting is this little meme here. The reason why it says not sure if indicative of ketosis or just bad breath, you see here when you have this conversion to ketone bodies, them traveling through the blood, going to muscles and tissues to convert back to acetyl CoA and back to get energy in the citric acid cycle. Where you notice with ketone bodies over here in the blood, when they're moving around, you also end up with ketone bodies in urine and you end up with acetone in your lungs from ketogenesis. Now, acetone, where have you heard of acetone before? Nail polish remover. So if you picture what nail polish remover smells like, and that's now in your lungs, so when you breathe out, that's why you end up with that bad breath kind of smell if you're doing a keto diet. Because these ketone bodies and their byproducts like acetone are now traveling throughout your blood and ending up in other places as well. Okay, so it ends up with ketogenesis. It's not just, okay, the liver mitochondria is going to take excess acetyl CoA, turn it into ketone bodies. They're going to travel to the blood, go to the muscles where I need and want my energy for my gains and working out. You forget that when it's going through the blood, it's also going to go other places as well. Okay, and so that's why you get those byproducts like bad breath. But overall, I always recommend to students don't do any of the diets that are fads and, cutting out whole groups or anything. In science, the best thing is balance. Do things in moderation. Really be aware of what you are putting in your body and don't trust fad diets because you'll be so focused on one thing, such as, oh, I want to lose weight or, oh, I want to gain muscle, that you forget that there's so much more, that there's a whole network, so many pathways, so much involved in the human body that you end up doing destruction to other parts of the body. And so, my biggest advice is whenever you are putting anything into your body or trying to make decisions, keep in mind your kidneys and your liver. A lot of the fad diets or supplements or things that people use to try and get more aesthetically pleasing, with weight loss or with muscle gain, they don't realize destroys parts of the body, including the liver, the kidneys, and the brain. And those are areas you don't want to destroy, and it puts a lot of strain on the heart as well. So be very careful with what you try. Okay, so now in biology, I've mentioned before, there's always, the opposite pathways that need to exist because, if you're going to break down fatty acids, you also need to be able to build them up when you need them. And so there are a couple things on this slide I want to point out. The first thing is the question that I have there, carbon substrates. When you are going to synthesize fatty acids and triacylglycerols, what are you going to use as the carbon source? And so you see right here to kind of help you remember, you derive them from dietary carbohydrates. So eating carbs will then send you onto the pathway that ultimately leads to fatty acid synthesis. Now, I want to point out, like I just mentioned a moment ago, when you have one process such as degradation, you need the opposite process in the body to balance it. So you need fatty acid synthesis. So in terms of comparison to degradation, I want you to think of these two processes as complementary to each other. And now notice we mentioned that fatty acid degradation, which we called fatty acid oxidation, that will occur in the mitochondria, whereas fatty acid synthesis will occur in the cytosol. And that's why we mentioned earlier the idea of the carnitine transport in order to get into the mitochondria, and you block that transport if instead you want to have fatty acid synthesis. Okay, so fatty acid synthesis will occur in the cytosol. Now, when it comes to fatty acid synthesis, I'm not going to make you memorize all of the structures that you see here. I just want you to know that it is a four-step process. You kind of saw this in the figures on the previous slide. Each step has the fancy enzyme name, such as beta-ketoacyl ACP synthase, beta-ketoacyl ACP reductase, beta-hydroxyacyl ACP dehydratase, and then enoyl ACP reductase. But you can simply think of the four steps as condensation, reduction, dehydration, where you see that water leaving, and then reduction again. Okay? And in reduction phases, you notice that it is guided by NADPH. So reduction, when you are looking at these processes, you're going to have, the electron acceptors, the electron donors that we've mentioned previously. And so you see in these cases, you have NADPH helping out with the reactions going on there. Now, another term that I want you to be aware of and circle star highlight is the term desaturating. So, specifically, desaturating enzymes, what will they be doing? Well, they will remove two hydrogen atoms from a fatty acid. When they remove the hydrogen atoms, they're now creating a carbon-carbon double bond. And what fatty acids have double bonds? Unsaturated fatty acids. Okay, so that is why they are called desaturating, because you are taking a saturated fatty acid and you are turning it into an unsaturated fatty acid because now it has the double bond here. Now, if you think of that, that's actually the opposite of something that we've talked about previously, which was hydrogenation. Okay, so the process of hydrogenation, you're going in the reverse. You're going to take that unsaturated, make it saturated, it becomes solid at room temperature, and now it's more unhealthy. Whereas desaturating, you're going the healthier route. You're taking the saturated, making it unsaturated. And when you think about that, think about what it does to that. You know, how it's now going to more likely be liquid at room temperature, have a lower melting point, okay? Just to help refresh your memory of some of the topics that we talked about before that end up coming back. Now, when we talk about lipids, remember we don't just have fatty acids as lipids, we also have other types of lipids, such as triacylglycerol. And if you remember, triacylglycerol is basically three fatty acids that have been linked to a glycerol molecule. Okay? And so, triacylglycerol synthesis, a couple of things to mention. First of all, you notice in this figure here, it's going to occur from what? From phosphatidic acid, which is right here. So circle star highlight phosphatidic acid. That's going to be your origin, like a cool superhero movie. The origin story of triacylglycerols is phosphatidic acid. And basically what is going to occur is dephosphorylation. And so you see that over here. So you're getting rid of a phosphate group, and then you are able to link on the third, the third fatty acid at the third carbon. Okay, because again, triacylglycerol, tri means three fatty acids. Okay, so you take that phosphatidic acid that has two fatty acid groups, you get rid of the phosphate over there, and then you link on a third fatty acid. Now, another really important form of lipid that we mentioned previously were the sphingolipids. And remember mentioning how myelin sheaths and neurons, the nerves, are very important to have sphingolipids with your nervous system. And so when we look at lipid metabolism, one of the things we have to consider is also the synthesis of sphingolipids, and they are derived from ceramides. Okay, so ceramides are the precursors for sphingolipids. You end up having ceramides produced in sphingolipid synthesis and degradation, and they're basically bioactive sphingolipids that end up being very important in terms of membranes and cell signaling. And so when you think of ceramides, think of them as helping to support the structure of your plasma membrane and the many cell signaling events that you have in eukaryotic cells. Now, when it comes to having lipid metabolites that accumulate, in terms of these processes and having ceramides accumulate, sometimes that can be a sign of heart problems. They use it as a prognostic indicator that you have some sort of cardiovascular disease. A lot of times they see it associated with people who have hypertension, heart failure, atherosclerosis, And so they'll take or they'll test your serum ceramide levels to kind of figure out if you've had any major adverse cardiovascular events. So it's kind of proof that something happened, something went wrong. And the reason why you see that accumulation in tissues is because there has been damage to blood vessels or the heart. And since the ceramides are critical structural parts of the membranes and these vessels, if there's damage to those structures, you'll kind of see the release of a lot of these ceramides in your serum. Um, in relation to them being part of membranes, that also brings us to their market use. So you might have seen lately a lot of areas suddenly popping up with beauty ceramides. And so in marketing, they've become part of beauty products because basically they can help hold skin together by forming what's basically a protective layer, and they can limit moisture loss, drying out, wrinkles from forming, and they can also help kind of protect against damage from pollution exposure, environmental stressors like, for instance, too much sun exposure. So basically, you see ceramides as helping to give you a protective layer on your skin in beauty products. Now, we already mentioned that a lot of what happens when it comes to metabolism is determined by whether there's high energy or low energy. And so, when we are looking at metabolism, for instance, when there is high energy in the cell, you don't want the citric acid cycle to run because you don't need more energy. The point of the citrate cycle is to get more energy, so we don't need that if there's high energy. So we have something called the citrate shuttle, and that will allow excess acetyl units to leave the mitochondria and go to the cytosol so that they could instead go toward fatty acid synthesis. Because when you have high energy, remember, you want to use that energy to build things. And one of the things you can build is fatty acids. Okay? So the citrate shuttle provides a way to get that excess acetyl units out of the mitochondria and into the cytosol. You could almost think of it as kind of the opposite of the carnitine shuttle, because that's getting fatty acids into the mitochondria for the breakdown of fatty acids, whereas citrate shuttle is to get extra acetyl units and citrate out of the mitochondria to go toward synthesis of fatty acids. Now, like everything else we've talked about, you always need to be able to regulate it. I said you don't want to picture the idea of a poor little cell exploding because it has too much product, too much synthesis. I know, a little dramatic, but you get the point. So, when it comes to the regulation of fatty acid synthesis, there are two main mechanisms of control. They will ultimately target the acetyl-CoA carboxylase that's involved in fatty acid synthesis. And the two main mechanisms are allosteric mechanisms and covalent mechanisms. So for allosteric mechanisms, you will see that over here. So citrate and palmitoyl-CoA can be controlled allosterically, meaning you have the idea of a binding to the enzyme, triggering a change in terms of its function, whether or not you will have it active or inactive. Over here, you see that the covalent modification that tends to be used to regulate fatty acid synthesis, you see that P symbol over there because it's phosphorylation and dephosphorylation. Okay, so you see when the enzyme is fully phosphorylated, it is inactive. And then you see over here, it becomes active once you get rid of the phosphorylation from it. okay, using phosphatases to regulate that enzyme. Now, one of the other important lipids that we previously talked about was cholesterol, and this is the one you hear all the time related to medical issues. Now, when it comes to cholesterol synthesis, you have it in all cells, but I want you to circle star highlight that it's primarily in the liver. Okay, the liver is the big location. Now, when you look at the synthesis, there are four distinct stages. But we've mentioned previously, whenever you have multiple stages in a pathway, the most regulated step is usually the first one. Okay, you can regulate the enzyme or the process in the first step to really control whether or not you get the final product. So to turn on or off that pathway. And so when it comes to cholesterol synthesis, I want you to circle star highlight stage one. Okay, formation of mevalonate. Make sure you know that word. Mevalonate is stage one of formation of cholesterol. The reason why I'm emphasizing that so much, and you see it over here as well, that synthesis of mevalonate, is because that's the big one in terms of regulation. That is a rate limiting step, and that's the step that we target when we give people drugs to reduce their cholesterol. So if someone has high cholesterol, you'll see again in later slides, we put them on drugs called statins, and the statins will inhibit this stage one. These statins inhibit or block mevalonate formation, which means you cannot go through the rest of the pathway, and so you get less formation of cholesterol. Now, ultimately, there are three main fates when it comes to cholesterol. Biosynthetic pathways can either, sorry, getting the laser, can either store cholesterol in what's called little lipid droplets. They can export the lipid into the circulatory system and get it to peripheral tissues, or it can be secreted into the small intestines through bile ducts. Okay, so these three squares demonstrate for you the three main possible fates for cholesterol once it's been synthesized. Now, the reason why we mention cholesterol so much, and we've touched upon this in the past, but this is an important review slide, is that cholesterol can be a major problem for your health. Okay, high cholesterol is a huge killer of people when it comes to cardiovascular disease. One of the big problems that you see occur is called atherosclerosis, and so that's demonstrated over here. You want your arteries to be nice and clear so that you can have proper blood flow. If you have high cholesterol, specifically the LDLs, remember the lousy, low density cholesterol, they accumulate along the walls of the arteries in the form of plaques. And that gradually narrows the blood flow. And you end up, if they get really narrow and hardened, you end up having increased risk of heart attack and of stroke. And it could be very problematic. So, whenever they detect that you have high cholesterol and things like exercise or diet are just not helping you, because again, we mentioned one of the things that can affect cholesterol can be genetics. There's a lot of factors. So, if your cholesterol is really high and lifestyle changes are not helping you, they will put you on statins. And the statins, like we just mentioned, those block the mevalonate synthesis. They block stage one of cholesterol metabolism, cholesterol synthesis. Okay, so those are very important drugs. Now, since we were just talking about cholesterol and the health complications, I threw in this slide that we've seen before in our previous lectures because I wanted to remind you of the types of lipoproteins The first time we looked at this slide, we started out by mentioning chylomicrons, which are the largest of the lipoproteins. They're ultra low density, which makes them very dangerous, and they basically transport triacylglycerides through your body, and that can be unhealthy. Now, the two that I told you previously to really emphasize and make sure you know the difference between are the HDLs and the LDLs. So high density lipoproteins, low density, but you can use the trick of H, think healthy, L, think lousy. The reason the HDL was healthy is that that is involved in transporting the cholesterol out of your body. So it brings it back to the liver to get exported and excreted out of the body, whereas LDL, what that does is it transports the cholesterol to get accumulated in plaques in the arteries, leading to the atherosclerosis that we just mentioned a moment ago. And again, if you remember, we've seen this figure as well to remind you the things that you'll want to avoid and the lifestyle changes that can help prevent you from having cholesterol problems. And if you remember, I highly recommended green tea on a regular basis for a million things. I call it magic. It's great for preventing or blocking allergies if you're someone who has seasonal allergies or, get bothered when someone has a pet around. Have green tea. It also helps prevent cancers and it is great for lowering your cholesterol. Also, omega foods like salmon, very good, or having kale on a regular basis, very good for maintaining healthy cholesterol levels. Then we get to this slide, and we're not going to go into the details of the mechanisms involved in this slide, but I will mention it because, as you know, anything to do with DNA or genetics, that's my baby. That's what makes me all warm and fuzzy on the inside. So I wanted to mention SREBP, which are sterol regulatory element binding proteins. And basically, I just want you to know that these are transcription factors that activate the genes involved in cholesterol synthesis. Okay, and they do it through a negative feedback mechanism. But it's always good to know when there are genes involved in something such as cholesterol synthesis, because then you can also use them as a mechanism to potentially develop drugs or treatments against things like high cholesterol. And that brings us to our review questions. You will be asked some calculation questions here. Also, keep in mind that a good way to practice is when you have review questions like this, where, especially where numbers are involved, practice more problems of it where you can just change the number given to you and boom, you have more practice. So here it says C14. Okay, try C15. Try C16 we did already. Try C18. Okay, so just randomly pick numbers and see if you can calculate. Another review problem. Quite a few review problems today. This is a great chapter for practice questions. For this one, make sure you also know the name of what is being blocked. That's an important aspect of statin drugs. And just a reminder, please make sure all of those review questions to send me your answers in the Remind app, just so that I can make sure that you're following along, understanding the topics, and to prevent you from making mistakes when you end up seeing similar questions on an exam, especially since some of this stuff comes back on standardized tests such as the MCAT. That is it for today. As always, thank you and have a great day.